We really have an exciting morning planned, actually I should say day planned, on AI, and I'm so glad you're all here to join us. I'd like to begin, though, by echoing Victor's thanks to those who have helped make this program possible, particularly Dr. Michael McGinnis, Dr. Se Kim, Sunita Christian, and Mizo Park. I also would like to particularly give a shout out to three of the committee members, Dr. Kevin Johnson, Patricia Brennan, and Peter Lee, all of whom were enormously helpful in helping in orchestrating today's program. Now, I have the pleasure of introducing our keynote speaker, Dr. Aviv Regev. So, Dr. Regev is head of Genentech Research and Early Development. Formerly, she was chair of the faculty and core member of the Broad Institute of MIT and Harvard, professor of biology at MIT, and a Howard Hughes investigator. She is founding co-chair of the Human Cell Atlas and has pioneered foundational experimental and computational methods in single-cell genomics, enabling greater understanding of cell and tissue functions. Dr. Regev is a member of the National Academy of Sciences, the National Academy of Medicine, American Academy of Arts and Sciences, and a fellow of the International Society of Computational Biology. I really refer you to our program so you can read about her many honors that she has received. I'd now like to welcome her to our podium to give our keynote address. We are really thrilled you're here with us today.
So, making medicines for patients is a series of multiple, very big problems across multiple scales. And when you think about the many different kinds of diseases, for example, breast cancer, IBD, lung fibrosis, you have to contend with the fact that there is a complex ecosystem of cells, disease cells and cells in the microenvironment. They affect each other through intricate molecular and physical interactions, and they're each themselves governed through complex molecular circuits inside them. And that means this complexity at every level. It means that we have an enormous problem with big numbers. There's more than 10,000 cell types, tens of thousands of genes, hundreds of thousands possibly of disease-associated loci, and more than 10 to the power of 19 ways in which they could hypothetically combine. And that's a lot, and it makes this collective mission, yeah, it's a lot. It makes this collective mission to understand and treat disease, rather difficult. And so what I'm going to try to describe to you today is how two parallel and mutually reinforcing and beneficial advances, on the one hand, a change in the nature of biological experiments, and on the other hand, algorithms and models and inferences of AI might take numbers that look like this, seem totally intractable, and put them within reach. And in order to understand how this can possibly happen, you will have to bear in mind a key principle. So for a long time, we thought that figuring out biology was a quality problem. You have to have a great way to look at everything with exceptional precision in one experiment. But as I'm going to show you today, when the quantity of data grows large enough, this quantity becomes a new quality. And so, when it's big enough, it's still smaller than these full huge numbers like 10 to the power of 19, but it's big enough to let AI models systematically figure out the rest for us. And to show you how this works out, I'm going to start by going to the fundamental unit of life, which is the cell. So in the early decades of genomics, we used to look at one tissue sample at a time. The sample would be made of many different cells, but we would look at all of them mixed together. If you think of every cell as a piece of fruit, we were doing the lab equivalent of a fruit smoothie. About a decade ago, we started developing new lab methods for single-cell genomics that allowed us to profile individual cells one at a time. And more recently, we moved to spatial genomics methods that allow us to profile the cells in situ in the tissues. Now, if you look at an advance like this through the lens of best quality, you would aim to make your lab methods focused on making a small number of very, very high-quality measurements of a few cells, but everything about those few cells. Instead, we chose to look at it as a quantity problem, and we developed a method for scaled data. These new lab devices, like this DropSeq device, microfluidics device here, where each cell is encapsulated in a droplet, and you flow maybe 5,000 cells in the device in 1 second. So we traded off here quality for quantity. So each cell's measurement is noisy and messy, but the number of cells that we can sample is many orders of magnitude more, and reliably, as a result, the scientific community has now profiled hundreds of millions of single human cells. But we made a trade-off between quantity and quality. And so the data are noisy and messy. And in order to make sense of data like these, we need AI algorithms. One unique quality of large-scale data is that they can let algorithms discover many different kinds of patterns, or as we call them, good representations. And when algorithms do this, the representations show us biological events. And so when algorithms cluster, we find cell types. When algorithms find continuous trajectories or transport maps, we see dynamical patterns like tumorigenesis or development. When algorithms find covariations across variables, we see gene programs, and so on, cell states, and even the physical location and the interactions between cells. So taking the large-scale data and the AI algorithm, we could assemble a human cell atlas and the associated disease atlases that now have more than 200 million cell profiles for more than 2,000 studies and tens of thousands of individuals. And in these data, algorithms help us make biological discoveries. So for example, in the cell atlas of the lung and the airways, we looked at the cells profiled from the trachea, and we discovered in them a new cell type that was never known before, right here, this little speckle over there that we call the ionocyte. And it turns out that this cell type, the pulmonary ionocyte, expresses and requires the gene that causes cystic fibrosis, CFTR. Still staying in the lungs, but moving to infectious disease, the Human Cell Atlas reference played a key role in the early understanding of COVID-19. First, in February 2020, very early in the pandemic, researchers used the reference atlas to show that the virus likely infects 82 cells in the lung based on the genes that those cells express. And using the cross-body atlas, we discovered cells all over the body that might be infected by the virus, in the nose, intestines, olfactory epithelium, the liver, the heart, the kidney, the brain, the vasculature, and more. And that helped explain the emerging pathology of severe COVID-19 at the time. Then we also created a dedicated atlas of cells from organs from autopsies of patients who succumbed to COVID-19 early in the pandemic. And algorithms analyzing this atlas helped understand the myriad of ways in which the virus can cause severe disease. Now turning to another example, in the cancer cell atlas of melanoma, an algorithm uncovered a particular gene program in cancer cells which leads to exclusion of T cells from the tumor and resistance to checkpoint inhibitor therapies. And even some treatment-naive patients already have these intrinsically resistant cells. So now this program can help predict which patients would benefit from a therapy and tailor their treatments. And also it discovers for us all these unique features in these resistant cells to target with medicines. And now if we turn to common complex diseases like inflammatory bowel disease, algorithms can link the differences between cells in the genes that they express with the genes that are associated through human genetics with the risk of disease. So here the algorithm used a colon cell atlas in healthy individuals and in patients with IBD to find cells whose expressed genes explain portions of IBD risk heritability. And once we know that, those cells become our starting points for new targets and specific diagnosis. Now, these data continue to grow rapidly. In fact, they've been doubling every 6 months for the past several years. Quantity became quality. Okay. So now, in addition to a healthy atlas from healthy tissues, there's a massive disease atlas, millions of cells from tumors, from COVID-19 patients, from patients with IBD, with cardiovascular disease, with dementias, and many, many others. But the more the data grow, the more laborious the analysis becomes, and it requires computational analysts, people to run all of these tools and methods. And also, the more demanding it becomes to really reason over all of this and make some unexpected discoveries. And this is where two other AI innovations come into play, autonomous agents and foundation models. So autonomous agents essentially are pieces of code, of software. They're equipped inside with an AI foundation model, like a large language model of human language. But they can do things. They can perform tasks, make decisions, take actions on their own, and they don't need continuous human guidance and supervision. An agent can understand the environment. The environment can be the world of computational tools. It can also be the wet lab. And it can make decisions based on this understanding. It can take actions to achieve specific goals. It has memory of past events and outcomes, and it can stop and interact either with the user either on its own volition or because the user interferes. And so agents can work at different levels of autonomy. On the one end of the spectrum, you have assistants. You're familiar with them because you use ChatGPT and other tools. And then on the other end of the spectrum, you have the fully autonomous ones. And an example of a general-purpose autonomous agent for biomedical research is this tool, BeyondMe. So we can hand BeyondMe a data set and a simple instruction. Perform annotation of this data set. And it's gonna do it in front of your eyes. It's gonna automatically make an analysis plan, and then it will set out and execute it. It will write the needed scripts. It will access the analysis tools and the files. It will run the analysis. It would look at the results. For example, here it ran clustering and differential expression analysis. It looks at the genes, and it decided to annotate the cells, annotated the cells, decided to plot the cell type composition, and finally, it's going to write for us a report with all of its insights. This helps tackle the sheer volume of the task. But all this agent did is know the things we already came up with, all these analysis tools and methods, and apply them in a new setting. But what about the analysis methods we haven't come up with yet? So as the data grow and grow, foundation models, like LLMs, can also help us query this atlas for new discoveries in new ways. So first of all, in these first atlases I showed you, the AI helped us find the answer that was already in the data that we measured. It usually also looked at one data set at a time, cancer, IBD, or healthy lung tissue. But with foundation models, it's different. We pre-train the model on all the atlas data from many tissues and diseases and contexts and even different modalities. It's not all RNA. But then we can use the model to perform tasks that we didn't train the model to do, pre-train the model to do. And we can also look for answers that were not in the measured data or the training data. And to illustrate this, I'm going to take you just through one task. So think of something like search. Imagine that you've identified one or a population of cells that you're interested in, and you want to search for similar cells, but without any specific annotation or clustering or signature. And until recently, no one really defined this task. I'm telling you that. Because largely, people were analyzing one data set at a time, so they weren't looking for anything. They were not doing searches. So how might we go about it? So I want you to think of this like reverse image search. You know how to run into someone in a meeting like this and you can't really place their face? Okay, here's what you do. You snap a quick photo, and then you run reverse image search and you save yourself the awful embarrassment. And so, the same idea, just with cells. Symmetric learning, as we call it. So we train an AI model with triplets of cells. Cell profiles, two of one type, one of a different type. Once we train the model, we bring in a query cell that we saw somewhere. It may be the kind of cell that the model never saw in the training, a new face. And yet, in a fraction of a second, here are other cells like it. The model can do this because it learned something general during the training about how to compare cells, just like Google Images can search for a face that it didn't train on. And so here's one of our searches. We start off with a particular profile of macrophages that were discovered in interstitial lung disease. And in these results, each of the blue circles is a tissue. The teal ones are specific diseases in that tissue. The orange ones are specific data sets. And they're scaled by the size, and they're colored by the fraction of cells that match the query. And as expected, you find fibrotic macrophages in many lung fibrotic diseases. But you also find them in COVID-19 samples, and in colon cancer, and in pancreatic cancer. So now I can focus on pancreatic cancer. For example, I can zoom into the data set, into the pancreatic cancer atlas, into these immune cells, and start asking questions about how they might contribute to disease. But now, things get complicated again, because each cell in itself is filled with these intracellular molecular circuits and all these genetic mechanisms that control them. And that means that the numbers are getting really big. So we need something. We need something more. So we already have data from cells in patients, and we already have AI algorithms and models that we can train and apply to find these good patterns. Now we need something more, which is systematic ways to perturb the system in order to find out how the system is wired and iterate. And this is what we call the lab in the loop. So for example, imagine that we wanted to learn an oracle that would be able to predict for any sequence of DNA of 80 letters, what level of gene expression would it drive? Let me tell you, there's 4 to the power of 80 possible sequences of length 80 over an alphabet of 3 letters. And if I made all of them and put them in a tube, its mass will be 20 times the mass of the Earth. That experiment is never going to happen. And so, what did people do in the past? They went for quality. They designed maybe up to 10,000 sequences that were based on their best understanding of biology. And the problem is that 10,000 is actually not a lot. And as a result, the models that you trained didn't predict well. But what if we went for quantity instead? Forget about designing sequences. Let's just test random ones, but lots of them. So, we made about 100 million random sequences. It's actually extremely easy to do in the lab. Then we put each of them in one cell. Then we measured how much expression the sequence generates. We then trained a new transformer model. This is the same architecture as large language models for human language, plus for DNA, on this data, and we used it as our oracle to choose new sequences and repeat it. And this oracle, this genie, performed exceptionally well at predicting the expression driven by any sequence, natural or random. So now, we use the oracle in order to design sequences that simply behaved in the way we wanted them to behave. The red sequences were designed to drive very high expression. The blue sequences were designed to give very low or no expression. This is real data, even though it looks like a cartoon. And that's exactly what they did, even more extreme than the natural ones in gray do. Okay, so now let's move from regulatory sequences to how genes affect each other to generate the cell's profile. Again, we use a loop. Here we start with primary immune cells. Then we use a method we developed called perturb-seq. We're going to use genetics with CRISPR in order to perturb individual genes one at a time. And then we're going to measure the effect on the profile of each cell for millions of individual cells by single-cell genomics. This means that in the sequencing, we will get the RNA profile of the cell and the nature of the perturbation in the same experiment. So we can do it in a very large pool. And then we learn a model of how the genes are wired that can predict what impact new perturbations will have, and then we can repeat the experiment. The model itself has 2 parts. On the one hand, it groups the perturbations into co-functional modules of perturbations that have a similar impact across the measured RNAs. On the other side, it groups the RNA effects into programs of genes that are co-regulated across the perturbations. And overall, this gives us a circuit. In this example of about 1,000 regulators in these immune dendritic cells, and how they connect to affect thousands of other genes across the entire life cycle of these cells. So this kind of perturb-seq loop, driven by AI, can be applied in many settings, not just to decipher regulation of pathways like I just showed you, but also to find targets that would give me the desired therapeutic phenotype in a cell, or to make me cell therapies that have desired properties, or in a co-culture or organoids to see how one cell type affects another, or combined with human genetic signal to find functions in human disease, or to help identify combinations of perturbations. But all of it, the atlas, the lab in the loop, they just looked at one phenotype in cells, just at RNA profiles. But when we try to understand a cell or a tissue by looking inside, We can do it in many different ways. Why just RNA? There's proteins, there's images, different components, resolutions, variables from molecular to visual, and each of them tells us something different about the tissue. But ultimately, we want to back out from these views into the holistic cell and the tissue. So can AI help with that? There's two reasons why that would be important for us. The first is it would allow us to model and understand the cell and the tissue causally and mechanistically, from the genotype to the cell state, interactions, niches, ecosystem, everything. The second is we just want to do more with less. Like, can we measure just one kind of thing and generate the rest of them with AI? So let me start with this utility first. I'm actually going to take you back to the big atlas for a moment. This is the cancer one. So obviously, it took enormous effort to collect this amount of data. And even though it looks like a lot, remember 10 to the power of 19, this is actually really little. So is there a way in which we can do more now without the need to continue doing all of these experiments? For example, what if I could go back to something really simple, like an H&E stain, which we have for almost every patient, and somehow just generate from it with AI, without experiments, these rich profiles, like single-cell RNA-seq or even a spatial protein stain? Well, there's AI for that. We developed an algorithm called SHAF that generates a tissue single-cell expression data set from the histology map. First, we need to train this kind of model. We can go about it in two ways. If we have training data where for every tumor I have an H&E stain and a spatial measurement, then I can train a model that can translate from each H&E pixel to the spatial measurement in that pixel. If, on the other end, I only have H&E and single-cell data of dissociated cells, then I can train a model that translates each H&E tile to single-cell RNA-seq for the cell at the center of the tile. And in inference, we take as input an H&E stain from a new tumor from a new patient. Paired SHAF is going to generate spatial transcriptomics data for the H&E, and unpaired SHAF is going to generate single-cell data, except that because it generates it per tile, it's going to be spatially resolved. You just need to think about it like translating from one language to another, the language of H&E to the language of molecular measurements. And we trained multiple such models, for example, a paired one for metastatic breast cancer and an unpaired one for small cell lung cancer. And when we apply them in inference, the paired model generated spatial transcriptomics data from an H&E of a new tumor of a new patient with metastatic breast cancer. And the unpaired one generated single-cell RNA data from a new patient, from the H&E of a new patient with a new tumor of small cell lung cancer. Okay, so now can we take this kind of approach into the inner wiring of the cells by combining them with perturbations? So for lab in the loop, we have a challenge. We have to match, remember, every perturbation with a phenotype. When we did it with Perturb-seq, we used sequencing. We sequenced the perturbation, its genetics, and we sequenced the phenotype. Turns out that's harder historically to do with imaging, because how would you sequence? And so, several years ago, Paul Bneny, actually one of my colleagues, developed a method called optical pool screens. You run your screen, you image what you want to image, and in the end, you use in situ sequencing under the microscope to see in each cell the identity of the perturbation. And last year, we added the ability to do this in primary cells and in tissues, and it's called PerturbView. So now, we have all the components. Can we do the corollary of SHAF in these cells? Do the same experiment twice. Some cells are going to be measured by RNA profiles. Some cells are going to be measured by imaging. Now, I just need an AI like SHAF to put them back together. And so that's what we did. We took some bone marrow-derived macrophages in this case, stimulated them with LPS and studied them, Perturb-seq, RNA profiles, Perturb-view, imaging phenotypes, the cell morphology, and 24-plex immunofluorescence. And so, because doing imaging is actually way cheaper and faster than doing all this sequencing, we did 3-fold more perturbations and 10-fold more cells with Perturb-view than Perturb-seq. And then we developed an AI method called Mokavi that allows us to put these two worlds together, just like the SHAF idea, but with perturbations. And after the AI puts them together, we can ask a very simple question. If I relate perturbations to each other based on their impacts, would I get the same or a different answer if I looked at RNA phenotypes or at imaging phenotypes? And it turns out they look really similar. Not identical, but really similar. Even when I zoom in here And look at this, you can see to the level of very fine processes. Okay, so now that we know that these two views relate so nicely, comes the fun part. Can we use the model not just to help us see what is in the data we measured, but like Chef, to generate the data that we didn't measure? That means, can I train a model to generate perturb-seq from perturb-view, RNA from images? And so in the training, I'm going to take as input these paired data set. Perturb-seq is small, perturb-view is large. I'm going to use Mokavi like before. I put them together. I already showed you that. In the inference, after I train the model, I'm going to ask the model to use the perturb-view data for which I've never measured RNA, only images, and generate the unmeasured perturb-seq profiles. And in this way, the model completed and generated the 2/3 of perturbations for which we've never done a profiling experiment. And when we zoom in, we see that the perturbed profiles that were generated by the AI, there are the ones in red, they group next to the measured profiles of related perturbations, which are in blue. And we also extended this idea to predict what might regulate in vivo macrophage states that I've not measured at all. They come from, not done at all in the lab, they come from patients. And so this ability to predict from partial data becomes critical when we turn to the biggest number, which is the number of combinations. So let's talk for a second about combinations. If I want to test experimentally all 5-way combinations over 20,000 genes, that would mean profiling 10 to the power of 21 cells. As a point of reference, each of your bodies is about 10 to the 13 cells. Not going to happen. I can make my problem smaller. I can look just at pairs. That would require 10 to the power of 10 cells. And of course, it's all just in one cell type in one condition. It's not everything in biology. If you want to be more modest, you can say, well, I only study immune cells and only the LPS response, and maybe there's about 600 genes that if I perturb them individually have a phenotype, so I only want to look at 180,000 pairs and only 18 million cells. But it's, it's more tractable, but it's still a lot, and still one cell type, one condition. But what if I used AI? What if instead of 180,000 pairs, I used AI to predict just the 600 pairs most informative to test in order to get a better model that can complete the rest? Those are, by the way, the very weak red dots that you can barely see. You need to have really great visual acuity for that. So that's what iterative lab in the loop is. So now we don't just start with cells, we also start with predictions. To select the first 600 combinations plus a slew of positive and negative controls, you collect the data, you feed it again to the AI. You generate another round of predictions based on the data that you measured. You feed the next 600 as predicted by AI, and you iterate. And you run through this a few times, not to 180,000, and you should get a model that predicts in general for this system. And so in this way, as we work iteratively through sequences or expression or images across systems and levels, we can build a foundation model of cell biology, or, as people like to call them now, virtual cells, that is a match to these massive numbers of biology. Okay, so now we have circuits inside cells, between cells, across the cells of a tissue, within a disease, in an atlas of many disease types across many patients. But can they actually help us with an ultimate goal of treating patients? With cancer or common disease or rare disease. And, maybe discover some new medicines for the patients. Like cancer genetics help discover specific oncogenic mutations, and then rational drug design help make molecules that target them, because that's the ultimate test. And as people in this room probably know a lot better than I do, it is a lot harder. So, not the least, because when you think about medicines, the numbers get really, really big. There's maybe 10 to the power of 60 small molecules with drug-like properties, and maybe 20 to the power of 32 different antibodies with therapeutic-like properties to consider. And in fact, every step in making medicines is hard, and all of those steps have to align in order to get success. You need the right target and medicine and dose for the right patients. And all of these things are not just time-consuming. Most of the time, the predictions end up wrong, not right. And so many of the great medicines that we have today rely on decades of biotechnological breakthroughs, recombinant DNA, antibody-based technologies, advances in chemistry, and so on. But as I just showed you, over the past decade or so, there's been an incredible pace of parallel breakthroughs in AI and in experimental biology that go hand in hand in this beautiful virtuous cycle. So, can this kind of loop between data from patients, AI, and experiments help us iterate our way to drug discovery and development? And so I'm going to close in the last couple of minutes with a super brief series of examples of what we're trying to do. So first come the targets, and how we want to try to work at those at very large scale so that we can quickly find more of these molecular circuits and use them to find more and better targets. You already know how it's done. You start with cells. You can screen them under hundreds of thousands of perturbations, genetic perturbations and small molecules. You can profile both the RNA and the images. I showed you how. You can put them together with AI. I showed you how. And as you do this, you can query the map for predictions that simultaneously finds targets and small molecule hits that generate the desired effect on the cell and become the starting point of drug discovery. And it's not just a cartoon. For example, in cancer, we pursue this type of approach in collaboration with Recursion. We conduct 2 loops in parallel, one in Recursion with cell images, one in Genentech with single-cell RNA-seq, and then we want to put them together in order to discover new pathways and new molecules to target them. Next question, indications. You want to span the full scope of opportunities, but also the full scope of liabilities for each target across the human body. So here we rely not on lab samples, but on patient samples to find all the relevant places where a target may be active and predict what may be the outcome of the intervention. Sorry, my clicker didn't click. Okay. In this way, AI helps us choose our target candidates for the maximal impact and specificity. Now, once we have a target, we have to generate the right medicines. These can be small molecules, antibodies, other proteins, vaccines, gene therapies, cell therapies. Each of them means a loop. For example, we use data from a large-scale screen in order to train a model encoder classifier that predicts for each molecule how potently it will act. Now we run a virtual screen for billions of molecules, using the model that we trained as an oracle to predict each molecule's activity. From these, the AI chooses new molecules to synthesize, which we test in the real lab, collect the data, improve the model, and repeat. And just like the example with the cells, here too, the AI is learning to generate the outcome of experiments we never measured. So it can predict new and more diverse molecules that end up as successful hits, and it does make us better. Using the same high-throughput data, the AI was about 50-fold better in the hit rate that it got compared to the original approach where an expert medicinal chemist made all of the choices alone on what to synthesize next. And it also got to new scaffolds that were not imagined by the expert. And we can generate the screen molecules in silico, not just for phenotypes like cell, live, and dead, but also against molecular targets, which we do with generative AI methods. And the exact same idea applies to therapeutic antibodies. Prior data is used to train the model. The model is an oracle. It is core many virtual designs. You make and test them in the lab and iterate. And when we run the loop for antibodies, we have to generate and score a lot of antibodies in silico, which we do by de novo diffusion models. Now, just like with single-cell analysis, these AI models can be used by scientists on their own, running all of these things and doing everything, or in partnership with an agent, like this friendly antibody optimization agent or your lab-in-the-loop agent. Once we have a potential medicine, comes the ultimate part, which is the patient. We want to identify those patients who will benefit the most from a medicine. And sometimes we have to personalize the medicine for the patient. And this is the case with cancer vaccines. Here we have a clinic in a loop. We sequence the DNA in a patient tumor. We identify the mutations. We use a model, which we trained on previous data, in order to predict which of these mutations is likely to trigger the immune system. The mutations become the basis of an individualized cancer vaccine. and made just for this patient. And this individualized cancer vaccine, or iNeST, which we developed in collaboration with BioNTech, is now in clinical trials. And for example, last year, Vinod Balachandran from Memorial Sloan Kettering published the results of a phase 1 trial in pancreatic cancer, where all the patients were treated with the vaccine, but those who had shown an immune response in red were relapse-free significantly longer than those in blue who did not show a response to the vaccine. And so, lab in a loop, clinic in a loop, foundation models, autonomous agents, drug discovery and development, all of these things work only when all components are tied together and they rely on everyone and everything working together, the lab, the clinic, the data, and the algorithms. But when they do work together, they have the potential to go after very big problems with very big numbers, even big ones like disease, and help us make more tractable for patients. Thank you very much.